Hi, I am Kalaivani Vetrivil from SNS College of Pharmacy and Health Science, and today let me talk about tricarboxylic acid cycle, that is TCA cycle. So TCA, tricarboxylic acid cycle, was named why citrate and isocitrate or tricarboxylic acids, and this was started with those two compounds. And it was also known as Krebs cycle by the name of a scientist Hans Adolf Krebs. Citric acid cycle, the first product of it was citrate. And nature of the pathway, it is amphibolic, that is both catabolic and anabolic pathways will be held, and the site of it is mitochondria, and it occurs only in aerobic conditions, and it also occurs both in fed and fasting state. So TCA is called a cycle, not a pathway, because it begins and ends with oxaloacetate. And coming to the amphibolic role of TCA, catabolic role of TCA, macromolecules like carbohydrates, fats, and proteins finally forms acetyl-CoA, which will enter into the TCA cycle. And anabolic rate role, many intermediates of TCA cycle synthesize important compounds and involve more enzymes. And this is the cycle of the TCA, and we'll go by step. The first step is condensation step, that is acetyl-CoA combines with oxaloacetate and gives citrate, that is citrate forms, and the enzyme involved was citrate synthase. Oxaloacetate is the first substrate of TCA cycle, and it is an irreversible step. Shall we raise the questions about oxaloacetate, whether it is a carrier of TCA and first substrate of TCA, catalytic role of the TCA? So we'll go into the further steps. We can get a conclusion that conversion of citrate to isocitrate. Citrate isomerized to isocitrate in two steps, that is dehydration to cis-aconitate and rehydration to isocitrate. And it is a reversible reaction. It is inhibited by fluoroacetate. Enzyme aconitase is a lyase over here. And the third step was conversion of isocitrate to alpha ketoglutarate. It is an 5-carbon atom, and isocitrate undergoes dehydration by isocitrate dehydrogenase to form oxalosuccinate, and oxalosuccinate is decarboxylated to alpha ketoglutarate, a 5-carbon atom, and in decarboxylation it requires magnesium and manganese ions, and first oxidative decarboxylation, and it gives rise to 1 NADH, and it is also a reversible reaction. And from alpha ketoglutarate to succinyl CoA, that is in fourth step, and alpha ketoglutarate 5-carbon oxidized to decarboxylated to succinyl CoA, that is a 4-carbon atom, and the second oxidative decarboxylation involves 1 NADH, and is formed here physiologically unidirectional step. Alpha ketoglutarate dehydrogenase inhibited by arsenate, and a multi-enzyme complex similar to pyruvate dehydrogenase, which need 5 coenzymes, and it was not regulated by phosphorylation and dephosphorylation, unlike pyruvate dehydrogenase. And finally, conversion of succinyl CoA to 4-carbon to succinate 4-carbon. That is the 4-carbon atoms present over here, and succinate thiokinase, the succinyl CoA synthetase, convert succinyl CoA to succinate. And the next is 1 ATP or 1 GTP is generated, that is in liver and kidney during the starvation, and this enzyme produces GTP. And the substrate level phosphorylations, these are the steps of the TCA cycle. And next, conversion of succinate to fumarate. So here involves dehydrogenation of succinate dehydrogenase, which will form fumarate, and FAD and iron sulfur proteins involved here over and was released and directly reduces ubiquinone in the electron transport chain. It is a part of a complex 2, and in TCA cycle, one intermolecular molecule will attach to the inner mitochondrial membrane, and succinate dehydrogenase inhibited by malonate. Here, conversion of fumarate to malate, that is by the hydration process. Addition of water across the double bond of fumarate forming malate, and enzyme fumarase is a lyase. Conversion of malate to oxaloacetate, that is final step in TCA cycle, converting the malate. This previously we said oxalate to oxalate. The enzyme is malate dehydrogenase, and malate is dehydrogenated to oxaloacetate, releasing 1-NADH. And oxaloacetate has a catalytic role like a homethane in urea cycle. So we can go for the regulation of TCA cycle. What will be the regulations? Citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase. Finally, we'll go for the energetics of TCA cycle. What are the energies will be produced? Means how much of energies is produced. The isocitrate dehydrogenase synthesis reaction, 1-NADH will enter into the electron transport chain, and 2.5 ATP molecules will be generated. And next is alpha ketoglutarate dehydrogenase. It is also 1-NADH will enter into the electron transport chain, and here too 2.5 ATP molecules will get generated. And the next step is succinate thiokinase reaction. In other is substrate level phosphorylation. The method is substrate level phosphorylation, and here 1 ATP molecule will be generated. Then succinate dehydrogenase, 1 FADH2 enter into the electron transport chain, and here 1.5 molecules of ATP will be generated. And malate dehydrogenase, 1-NADH enter ETC into the electron transport chain, and here 2.5 ATP molecules will be get generated. So finally the total number of ATP per turn of TCA cycles is 2.5, 2.5, 1.5, totally 10 ATP molecules will get generated. And again, energy yield from one molecule of glucose under aerobic conditions. It is from aerobic glycolysis, 7 ATPs will be get generated, and from pyruvate dehydrogenase, as 2 pyruvates from one molecule of glucose will be get nearly 5 molecules of ATPs and 2 NADH. And next is from citric acid cycle, that is 2 acetyl-CoA from one molecule of glucose. Here we'll be getting 20 ATPs, that is 2 into 10, 20 ATPs, and net ATP from one molecule of glucose under the aerobic condition is 32 ATPs, that is 7 plus 5 plus 20, so total 32 ATPs. And the net ATPs from one molecule of glucose under anaerobic condition is only 1 ATP molecules, or sorry, 2 ATP molecules will be get generated. So thank you for your attention.